I took a yogurt container and cut off the bottom, and then put a little LED in it and some light sensors, and I strapped it to my face and reflected light off of my eye, and then used that to track which way my eye was going. And then I built a little circuit to mimic communications of the mouse to the computer, and so that way I could control the cursor with my eyes.
Hey there, I'm Luca Fusar-Bassini. I'm a PhD student in computational biology at EPFL in Switzerland, and you're listening to The Biotech Futurist. The Biotech Futurist aims to foster deep understanding and discussion about exciting hot topics in biotech, but I want to say from the beginning that it is by no means rigorous in teaching the subject. And for the sake of outreach, sometimes we need generalizations that, of course, simplify the reality of the science behind what we're discussing. But I can say that my guests and I do our best to be clear and to go in depth. You can imagine to be out with me and my expert guest for a friendly conversation, to get a general understanding and more curiosity, having fun as much as I've had recording this podcast. This podcast has no sponsors, and any reference is not meant to support any commercial activity. This podcast is a solo effort, so if you wish to support me, I'd be grateful if you followed the Biotech Futurist on Spotify, Apple Podcasts, YouTube, Instagram, or your top podcasting platform, and share it with your friends. With that said, I am excited to move on to today's conversation at the Biotech Futurist. Adam Cohen is a professor of chemistry, chemical biology, and physics at Harvard. Quoting his webpage, Adam's lab works on understanding and controlling light-matter interaction in warm, wet, squishy environments. As you probably know, optogenetics aims at controlling neuronal activity with light. Well, Adam's work has worked out the other way around, so to record neuronal activity by converting electrical signals to fluorescent flashes by engineering a gene from a dead sea microorganism. And well, his research goes well beyond this. In 2007, aged 28, Adam was nominated by the MIT Technology Review to the 35 World's Top Innovators Under 35. Adam, I'd be very happy if you could tell us your story, scientific dreams, and achievements in like 5 minutes, please. Hard question.
Well, thank you for the invitation, Luca, and it's nice to meet you. Thanks to the listeners for joining. My name's Adam Cohen. I grew up in New York City. As a child, I became interested in electronics and circuitry, building things. When I would walk down the streets of New York, I would find televisions and computers in the garbage, and I used to take them home to take them apart to see how they worked. And I set up my bedroom in Manhattan as a little electronics shop. And then when I got to college, I became interested in studying physics and chemistry. And I worked in the lab of George Whitesides, who is a professor here in the chemistry department, on the topic of molecular electronics, which at the time was a idea that maybe we could replace silicon with molecular scale devices to make even smaller and faster computers. Now, of course, today we're still using silicon chips, and so as a technology that didn't really work out, but it was very interesting to study the electrical properties of molecules, and there are descendants of these ideas in many of the organic LEDs, for instance, that are in today. After my undergraduate work, I went to Cambridge, UK, to study there. And initially I was interested in some bio-nanotechnology areas, but for a variety of reasons, I ended up working on doing theoretical work. And I studied the theory of the interactions of light with matter and how optical forces could affect the mechanical forces between molecules and could affect the properties of matter. I did that for a few years and then decided I really loved building things and tinkering with my fingers, actually making physical devices. So then I went to Stanford and joined the physics department there. And joined the lab of W.E. Murner, who is a professor in the chemistry department. And there I worked on a project to build a machine to trap and manipulate individual molecules in solution. So you might know that if you have a molecule in water, the molecule can jiggle around randomly from Brownian motion. All the water molecules keep banging into it, and it moves around. It's a little bit like a classroom of 5-year-olds, all the molecules going every which way. And this makes it hard to study the individual molecules because they don't hold still. So for my thesis work at Stanford, I built a machine which could suppress the Brownian motion of an individual molecule. It worked by using a laser system to track the random motions of the molecule. And then there was a feedback loop that applied electric fields to the solution to induce an electrophoretic motion that canceled the Brownian motion. So this is a little bit like balancing a broom pole on the palm of your hand. And whichever one, just push it back in the opposite direction to keep the molecule fixed. And this would let us then suspend a single molecule in solution and then study its optical properties for a while, without the molecule running away. So that was a lot of fun, and I spent about 4 years working on that machine and demonstrating its couple of uses of it. And then I started my lab at Harvard in 2007 in the chemistry department here. And I vividly remember on the first day walking in, and it was just an empty concrete shell of a space. There was nothing there. And thinking to myself, oh no, what am I going to do? I have to do something. I really had no idea what I was going to do in my lab here. And so for the first few years, we explored a bunch of different areas. We studied effects of magnetic fields on chemistry, so whether really small magnetic fields could affect chemical reactions. We studied the interaction of light with chiral molecules, which have a handedness, where the molecule and its mirror image are not the same. And then more or less by dumb luck, we stumbled onto these proteins that come from a Dead Sea microorganism and can convert the electrical activity of a cell into changes in fluorescence that you can see in a microscope. And this came out of some basic spectroscopic studies. And at the time, I hadn't really studied much neuroscience, but I had a vague notion that neuroscience was a thing that people cared about, and that there was interest in the electrical activity of cells. Then over the last basically decade, or maybe even a little bit more, my lab has gradually shifted towards developing these tools and applying them to study different aspects of brain function. And then we became generally interested in understanding brain function and have now started to develop other tools as well to try to interrogate the activity of neurons and other cell types in the brain.
Mm-hmm. Using nice and curious proteins. And nice and curious proteins will be the main topic of today's podcast. Briefly before jumping to the main topic, I'd like also to ask you if you can spend a couple of words about your Wikipedia level inventions at high school. So you've been a creator and then inventor since all times, I think.
Sure. So as I mentioned, when I was in high school, I interested in electronics and building different devices. And one of the first big projects I undertook was I built an eye tracker for my computer. So this was I took a yogurt container and cut off the bottom and then put a little LED in it and some light sensors, and I strapped it to my face and reflected light off of my eye, and then used that to track which way my eye was going. And then I built a little circuit to mimic communications of the mouse to the computer, and so that way I could control the cursor with my eyes. Anyway, that was a fun project. And then the biggest project I worked on in high school is I built a device called a scanning tunneling microscope. This is a kind of microscope which can see individual atoms on the surface of a material. And I came to this because I had thought that electron microscopes were, these incredibly big and complicated and expensive machines which required vacuum and high voltage and would be totally out of reach. And then when I was in 11th grade, I visited Princeton, and I saw that in one of the undergraduate physics labs, this thing called a scanning tunneling microscope, which the students were using, and it was actually quite small. It was about the size of a microwave oven. And so I thought, if it's that small, it can't be so hard to build. The way a scanning tunneling microscope works is you take a very sharp wire, and if you cut the wire just right, there's one atom which pokes out on the tip a little bit more than all the other atoms. And if you then take that wire and you very carefully manipulate it to bring it close to a conducting surface, if the wire gets close enough to the surface, the quantum mechanical wave function of the atoms, of the electrons in the wire will overlap with the wave function of the atoms on the surface, and you can pass a tiny electric current from the wire to the surface. And that electric current gets bigger if the wire gets closer and smaller if the wire gets further away. And so then if you very carefully move the wire over the surface in a raster pattern, you can map the current and you can map the surface of the sample. And so you can make a map of where the atoms are on the surface of the sample.
Yeah, that's
and
inspiring.
So I thought that sounded really cool.
Yeah, that is.
It's a system that the people who invented this in the early 1980s got the Nobel Prize, I think in 1986, for inventing this machine. And so I decided to build my own. And so I made a structure out of Legos to hold all the parts together. And then I used some piezoelectric speakers for doing the fine manipulations, and built some electronics. I hacked the sound card from my computer as the analog to digital converter for the digitization. And so eventually I got this to work. It didn't work that well, but it worked well enough. Some of the challenges with this, there's the vibrations, because of course you need to position the wire relative to the sample with atomic precision. And in New York City, we lived up on the 18th floor of an apartment building next to a big highway and next to a heliport. And so of course, there's a huge amount of vibrations in a building like that. And so one day when my parents weren't home, I drilled a hole in our ceiling and suspended the whole system from elastic bungee cords hanging from the ceiling using some heavy biology textbooks as ballast. So then this whole thing was hanging in the middle of my room, and that's how I isolated it from the building vibrations.
That's creative.
Anyways, that was a lot of fun to build.
I guess, guys, now we have understood that Adam is a big inventor, so maybe we can jump to the inventor, which is the main topic of this podcast today. So let's just take cells. Well, cells can themselves take selfies. You know, there are now ways to record the story of single cells from the inside. And Adam has been working on a very cool project. And I think it is still a preprint entitled Time Ticker Tapes for Intracellular Recordings. Right? So, Adam, you built an incredible system based on self-assembling proteins to create a timestamp of cellular events. But other people are trying to do so with nucleic acids. Can you briefly let us understand the system and explain why proteins and non-nucleic acids? I mean, proteins for sure have the advantage to enable real-time monitoring when one uses fluorescent tags, but they have a hard time at multiplexing, whereas CRISPR arrays look more straightforward to my eyes of a student. So yeah, I have a ton of questions regarding this, but maybe first you can start introducing how this works and why proteins.
Sure. Yeah, so first I'll just tell you about the system we made, and then we can discuss the relative features of this relative to the DNA arrays. So the system that we built is inspired by the phenomenon of tree rings in a tree. So if you go into a forest and you take a cross-section through a tree, you can see these rings representing the annual growth of the tree. And you can look back sometimes over hundreds of years, and you can learn about the climate and other things that happen each year just by counting the tree rings and then looking at the growth during that year. And so I thought maybe we could do something similar in cells. If we could get the cell to produce some structure which grows continuously over time. And then we can put marks in that structure which reflect different aspects in the cell's life. Maybe we can have a permanent record of some aspect of the cell's biology or experience. And so a fantastic postdoc in my lab named Ding-Chang Lin started to look for proteins that would grow and would have useful properties. Ding-Chang is now an assistant professor at Johns Hopkins University. He just started his lab a few months ago. And so he looked around in the literature for different proteins, and he came across this amazing paper which described a protein called PAC4. It's a protein that's some kinase. Its native biological function is not that important. But it turns out that if you produce a lot of this protein in a cell, it crystallizes into these fibers which grow from their ends. And so as they grow, this fiber gets longer and longer, a little bit like a knitting needle inside of the cell. And one of the neat features of this protein's structure, the crystal structure, is that there are pores which run the length of the fibers. And there's a lot of space in these pores. They're about 8 nanometers across, which is pretty big. So there's these pores, and you can attach different payloads to this PAC4 protein, and they'll get incorporated into the pore. And so as they grow, whatever's on the PAC4 protein will get incorporated into the pore. And so I thought that this could be a promising approach for recording cellular histories. Now, one of the challenges with this approach is that, of course, every cell is going to be a little bit different, and these proteins, these fibers, will grow at different speeds in different cells. And so we need something that's like the seasons, some way of marking the passage of time in the real world on these fibers, so that we can then connect events that we see on the fibers to the passage of time. And there's many ways you might imagine doing this. We initially have followed a fairly simple approach, which is as follows. There's a very neat protein called the halo tag, which is a small protein that's not by itself fluorescent. But this protein can form a chemical bond with a particular chemical motif. And you can attach that motif to fluorescent dyes of many different colors. And so what we do, and these dyes can go into a cell and get washed out relatively easily. So we fuse the halo tag protein to the PAC4 scaffold. We then add to the cells different colors of dyes at different times. So every couple of hours, we change to a new dye, which is a different color. That dye then labels, it binds to the halo tag proteins and gets incorporated into the scaffold. It is a series of stripes on this growing fiber, where each stripe corresponds to a different dye color that we've added. We know exactly when we added the dyes, because we look at the watch, we look at the clock when we do it. And so we know that the boundaries between the stripes correspond to specific times.
Exactly. So let me know if I understand correctly. For now, we are at a validation phase where we have this system that can basically record time, and we make sure that this time is consistent for a given cell type to the sequence of dyes that we give to the cells to record time. Now we want to make this a recorder of cellular events, right?
That's right. Yeah. So far, all we've done is we've set up our metronome, and so we have these marks which indicate every couple of hours exactly what time it is, or what time on this growing fiber. Then we want to now encode some information on this ticker tape that we've created. And there are a variety of ways you might imagine doing it. What we've done so far is we've used what's called a promoter in the cell. So this is a piece of DNA which can be activated in response to some aspect of the cellular environment or the cellular physiology. And we use that promoter to drive production of a PAC4 scaffold fused to a fluorescent protein tag, which is a different color from all of the dyes that we used for making our timestamps. And so when that PAC4 fused to GFP, for example, green fluorescent protein, when that is produced, it gets incorporated into the fibers, and we get green stripes in the fibers. By recording where those green stripes are relative to our fluorescent dye timestamps, we can learn the history of activity of the particular promoter that we picked. And so with that scheme, we picked a promoter called the CFOS promoter. So it's a promoter that gets turned on when neurons are active. So every time you think about your breakfast or whatever you're doing, some neurons in your brain become active, and then your neurons need to change which genes are expressed depending upon their level of activity. And CFOS is one of the ways that neurons do this. And so we borrowed that CFOS promoter. And so now when the neurons are active, that leads to green stripes. And so we can record the history of neural activity in the patterns of green stripes in these fibers. And that's where we are today. If you like, we can now talk about comparing this to the DNA-based ticker tapes, and we can talk about potential future directions as well for some of these protein-based systems.
Sure. First, a quick question. Do you try this system also in other cell types? And I think you have ambition to try this then in living systems. So first cell types and then living systems, maybe?
Sure. Yeah. So we've tried it in a couple of cell types. We tried it in neurons, and we tried it in a human cell line called HEC cells. These are cells which we just grow in the lab. And it works in both of those. We haven't yet done a broad survey of lots of different cell types, although I think that could be an interesting thing to do. Ultimately, as you say, our dream is to do this in live animals. And this gets to one of the challenges that we've encountered with the system that we have right now. These fibers that we grow are stiff, and when they first start to grow, they undergo a period of very rapid growth, which gets them quite long. And the fibers get so long that they're larger than the cell that they are growing in. And so they end up deforming the membrane of the cell. And so in the cultured cells, it can end up, it looks a little bit like a hamster that swallowed a knitting needle or something, where you have these fibers which can extend tens or even hundreds of microns beyond the normal boundaries of the cell. Surprisingly, the cells seem not to be so much bothered by this. The membrane just deforms around the fiber, and the fiber is still entirely within the cell, but the cell has this sort of long tube of membrane.
I've seen that you had data showing that neurons work well and the electric properties of neurons are not touched by this.
Yeah, the neurons seem to be fine, and it might be so surprising if you think about neurons have dendrites and axons, which can extend for centimeters or even meters in people away from the cell body. And so neurons are quite comfortable having long membrane tubes which point out in different directions. And so this is just another membrane tube which pokes out from the cell. This is in cultured cells. Now, in a live brain, I imagine that it wouldn't be so great for the animal to have a bunch of knitting needles penetrating through its brain. And so one of the challenges which we're working on solving right now is to better control the nucleation and growth of these fibers so that they grow in a more controlled way, so they don't deform the cell as much. And then we're also thinking about other potential scaffold materials which might either be flexible, so that when they get to the side of the cell, they bend rather than deforming the cell, or be in the form of a helix, so that you can get a lot of length inside of the cell without bumping into the boundaries. Or there's a variety of different geometries you could imagine, which might be a little bit gentler inside of a brain. I should say that at the same time that we did, there was another lab here in Boston led by Ed Boyden, who's at MIT, and his lab was working on a similar idea. They came up with a different protein scaffold, and it has some different technical properties, but they were able to demonstrate that they could grow these scaffolds in the brains of live mice.
Ooh, that's big. So there are different technologies going towards the same aim, I know, and that's beautiful. And yeah, maybe they're also good to multiplex and to learn from one another. So I think it's time now to compare your technology and in general protein-based technology to DNA and RNA-based arrays, especially CRISPR-based technologies that can record cellular activity. I mean that these protein level technologies are great because they can have a fluorescent readout, for instance. And as you have discussed until now, they are pretty straightforward to interpret, I guess, while CRISPR arrays maybe require a bit more of computational demultiplexing. But CRISPR arrays, to the eyes of a student, also look better to multiplex to record more events in parallel in a cell, while protein-based arrays are still limited by the fluorescent spectrum. So how do you think this system can multiplex, or is it just made for single events and for multiplexing something else is needed? How do you compare to RNA and DNA-based technologies?
Great. So it's an interesting question. So people have been thinking about different recording schemes in cells for a long time. And just recently, in parallel with the work we were doing, several groups have been trying to develop nucleic acid-based recording systems. And most recently, there was a beautiful paper in Nature by Jay Shinder's lab, where they made a CRISPR-based system. So since that's not my lab's work, I'll talk about it a little bit, but you'll have to forgive me in advance if I mischaracterize it or don't get all the details exactly right. The scheme basically is, so CRISPR is this amazing new gene editing technology, and there's many different flavors which can do different kinds of transformations. This DNA typewriter concept, the way it works is that the CRISPR needs to recognize a particular motif on the DNA in order to do its editing. And, or not the CRISPR, the Cas protein. And the system is set up so that the recognition motif on the DNA is only, there's one complete recognition motif, and then after it, a series of partial ones, which are not yet ready to be recognized. And the Cas editor comes in and introduces a sort of code into the DNA, and then prepares the next recognition site to be edited. And so then when that one gets edited, that then in some sense unmasks or reveals the next recognition site, and so on. And so it's like dominoes, where each one that gets edited then prepares the next one after it to get edited. And so this is a way of ensuring that you get sequential editing. And so there's two parts step. One is unmasking the next stage, and the other is incorporating a little DNA sequence, which has some unique information in it, which is like a barcode, which tells you something about the nature of the event that's getting encoded. It has some information. And so you refer to the ability to multiplex, and the multiplexing is this capability to write different sequences in the DNA. And so they call this a DNA typewriter, and that's a pretty good analogy because it puts different pieces of information in a unique sequence on the DNA. So that's super cool. And we used, for instance, for lineage tracing or perhaps for recording other aspects of the history of gene expression in the cell. Okay, so what are the relative merits of the protein-based and the DNA-based systems? One difference is that the protein-based systems, the readout is by microscopy. And so it's done in real space in the intact sample. And so you read the history of a cell and you know exactly where that cell was. You can look at who its neighbors are. You can look at its shape. You can stain it for other markers. You can learn anything else you want. In contrast, the nucleic acid-based systems, the readout is sequencing. And so this is typically done by grinding up the sample or mushing it up somehow, and then reading out the sequences. And so you get the sequence information, but you've lost the information about where that cell was or about anything else about that cell. Another difference is that the ticker tape system that we develop based on proteins has an absolute time base. So we know not just the order in which events occurred in the cell, but the actual time at which they occurred. And it's an analog system. So you can see how much or little of the protein that you're interested in was incorporated into the cell. And you can even map the time course of its activity by looking at fluorescence as a function of position along this fiber.
That's great. And yeah, you can also do so for many cells in parallel and with a relative spatial position, I guess.
That's right. The nucleic acid-based systems are binary. So either the gene was edited or it wasn't. And you don't have any absolute timing information. So you only learn about the order in which events occurred. So that's in terms of the systems which have been created sort of as they are right now. In terms of future uses, one of the things that I'm excited about for the protein-based systems is to make the incorporation of fluorescent tags conditional on things other than gene expression. So right now, for both the DNA-based systems and the protein-based systems, all we're measuring is activation of different promoters in the cell, turning on a different gene expression program. But of course, cells' lives is much more than just the genes that they express. So, neurons fire and have calcium. There's all sorts of signaling, kinases, phosphatases, lots of different enzymes that do things in the cell that don't go through transcriptional pathways. And proteins are these amazing multifunctional tools. And it's at least conceptually possible that you could make the incorporation of different fluorescent marks in a fiber conditional on things like the calcium level in a cell or the activity of a particular phosphatase or kinase or some other biochemical signal. And so I think there's a possibility to generalize to other kinds of information and other kinds of modalities beyond gene expression. And that's not something we've done yet, but that's a direction we're going in. I think will be interesting and distinct from what would be possible with a nucleic acid-based barcoding system.
That's wonderful, but certainly inspiring. So staying with our proteins, how far can multiplexing get according to your vision? And how many colors or how many different types of structure? What are the ideas to enable a bigger multiplexing? Yeah.
Sure. So by multiplexing, I suppose you mean like how many different genes you could monitor at the same time, or how many different transcripts you could monitor at the same time. Yeah, so this is pure speculation because we haven't done this yet. But you could certainly imagine having Pac4 proteins with different peptide barcodes on them. And of course, of different peptides that you could put on the protein. And then the question becomes, okay, how many of those can you read out? If you're going to try to read them all at the same time by fluorescence, then as you hinted, we're limited by the number of colors you can distinguish at any one time. And depending upon how fancy you are with your lasers and with your microscope, somewhere 5 to is the spectrum gets pretty crowded beyond that. But I think it's conceivable that you might be able to go further than that. And here's one example of how you might do it. There are thousands of different antibodies, each of which recognizes a different particular peptide protein motif. That's called an epitope. And so and now there are systems for sequentially staining with different antibodies and then washing it off and then staining with a new one. And so if you can stabilize these fibers enough so that you can stain with one antibody and then wash it off, and then stain with another and then wash it off, and do that over and over again, then in principle, there's no upper limit on the number of different antibodies that you could stain with. And as long as you're patient enough to keep doing this over and over again, you could read out as many different epitope barcodes as you wanted. I mean, that's just a concept, we haven't done it yet. And I'm sure there will be some devils in the details, getting antibodies in and out and having the system be stable and so on. But it's not a totally preposterous thing to try.
Yeah, that's great. And what about time resolution?
I mean, one of the limitations is that for each thing you want to record, I should say one of the limitations is both for this and for the DNA barcodes is that for each thing you want to record, you have to introduce a new genetic element into the cell. And at some point it becomes cumbersome to introduce 10 or 20 different payloads into cells. The gene delivery efficiency is not 100%. And so if you're trying to do a large number of different genes, the chance of a cell getting all of them starts to become rather small.
Molecular biology issues. So
yeah, that's right.
My next question would be, how can you increase time resolution? I read in the paper that now we can record events separate by like 40 minutes or something like that. And how can you, for instance, tune the half-life, as you mentioned in the paper?
Yeah, so it's a great question. And there are several aspects to the time question. One issue is if the fibers grow faster, then the time stamp marks become further apart and we'll get better time resolution. But then, of course, they're going to bump into the edges of the cell. And so there's a trade-off between time resolution and total recording duration, dynamic range. And so, for instance, if you want to record for days or weeks, then you would probably have lower time resolution than if you're willing to just record for an hour. And so one thing we're looking into is ways to control the growth rate of these fibers so that we can try to control the time resolution. If we had thinner fibers where we could have a much longer fiber inside the cell, then it could both grow fast and we could have a very, very long recording as well. So that would be a wonderful thing to do. Another aspect of controlling the time resolution is controlling the interval between your tick marks, between the seasons or the timestamps on these fibers. As you can imagine, if you have a clock which dings once an hour, it's harder to know exactly when something happens compared to if you have a clock which is giving you a time every second, right? And so right now, the timing between our timestamp marks is set by the time to wash in and wash out these fluorescent dyes that we're adding to mark time. We're thinking about systems where you can make a mark of time with just a flash of light using some light activated.
Also for living animals, I guess it's not so suitable to give them dyes.
Well, it turns out the dyes, you can just inject them into the bloodstream and they cross the blood-brain barrier. And
Isn't that
you
slow?
could do periodic. It's actually not that slow. A couple minutes.
Oh, wonderful. That's nice.
Dyes,
wow.
To get in. Yeah. But of course, as a reagent, photons can enter and exit a sample as fast as you could hope. And so, the photons don't hang around. You don't have to wash them out. And so as a tool for making timestamps, I think photons would be a very attractive tool. Of course, with applications in a live animal, then you have to think about how you get that light into the target tissue.
Yeah.
And so ideally you would use red or even infrared light, which penetrates much better into tissue than bluer wavelengths of light.
Your passion to light is pretty clear. So
yeah. I mean, photons are an amazing reagent for doing biochemistry.
I'm also curious how you decided at first how to overexpress to a given amount the protein in order for it to grow quick enough but slow enough. You know, if you overexpress the protein more, probably the growth would be uncontrollable, or if you express the protein at too low level, maybe the resolution will be too bad. How do you do experiments to decide the expression level, and how do you control it?
Yeah, so that's a great question, right? I mean, the way we started was very simple. We had cells growing in a series of wells, and we just did a concentration series on the plasmid for the ticker tapes, from very high concentration to very low concentration, and then we looked at the different wells to see which ones looked best. One of the challenges is that even for a given nominal dose of the plasmid that carries this ticker tape protein on it, the scaffold protein, there's tremendous variation between cells in the expression level. So some will express a lot and some will express none at all. And that's because different cells will take up different amounts of plasmid, and there's a whole bunch of sources of variation in the population. So one of the things that we're thinking about is how to get a tighter distribution of the expression levels within a population of nominally similar cells. And this is a problem many people have worked on. There's, for instance, feedback systems where you have, in addition to the protein you're trying to express, you have it express an inhibitor of its own promoter, so that if the expression gets too high, it shuts itself down a little bit. So it's a little bit of a self-regulating expression. There's also different gene delivery techniques, whether you do transfection or lentivirus, or you make a transgenic cell line, which have different levels of variability in the expression. Finally, to just control the absolute expression level, there are what are called drug-induced promoters. So these are promoters, which can control production of RNA and then a protein, where the activity of the promoter is controlled by some drug, and you try to pick a drug which you hope doesn't do too much else to the cells or to the creature you're giving it to. And so then by controlling the time drug, you can control when the expression comes on and how much it comes on. And so that gives an independent degree of control.
That's interesting. Yeah. My next question is, what type of information can this system provide in addition to what we can get from omics? You know, all the time record has something about what happened in the past and what's happening now, so you can compare for sure, something that with omics you can only do indirectly. Do you have any suggestion on what we can do with this system that omics cannot do?
Sure. I mean, there's many different omic technologies, and so I'm not sure exactly which one you mean. I mean, there's proteomics, there's transcriptomics, but I mean, I think you mean generally technologies for profiling some molecular markers across the tissue. And I mean, those measurements are usually endpoint measurements. So to get your molecular markers out of the tissue, you have to kill it and extract them, and they'll tell you something about the state of the tissue at the time you measured it. With the tools that we're developing, you can, we hope, learn about the history of the tissue. So learn, not just what is it doing now, but for each particular cell, what was its life history in some along some dimension, by some measure, and so that can tell you perhaps about dynamics. So and you can record things that are not big macromolecules. So at least in principle, you could, for instance, record a history of calcium activity, which is something that disappears the moment you kill the tissue, but is much closer to the underlying neural activity and could be a record of neural activation. Again, we haven't done that, but that's something we're
Yeah, I was
hoping.
also thinking of like a ligand binding a receptor or a kinase activating, something like that.
For sure. Yeah, all GPCR signaling, receptor tyrosine kinase signaling. There's a ton of stuff which goes on in the cells, and I think in the omics world, people tend to have a very sort of perhaps focused or myopic view of cellular physiology. But you have to remember, cells are a lot more than just transcription and translation, right? There's all of this signaling which happens. And particularly for things like neurons, which have to respond quickly, most of the interesting dynamics don't go through transcription.
Yeah, they are so hard to capture. Related to this, what is a kind of application that you envision will happen soon or that you are working on to use this system to understand a biological problem or to answer a biological question?
So my dream, my dream, which is what motivated me to really start this project, is to try to understand something about the nature of memory. So if you think back to what you had for breakfast this morning, presumably everybody can remember what you had for your breakfast. And so there's some physical embodiment in your brain, some three-dimensional representation of that memory inside of the neurons in your brain. But we have no idea what that representation is, how that information is onto the neurons in your brain. And so my hope is that by recording the history of neural activity in an animal that is going through different environments and learning different things from each of them, we might be able to see which subsets of neurons were activated in each environment. Then we can test the animal's memory or recollection. We can test its forgetting of these different environments or different contexts, and perhaps learn something about how those how that information is represented inside the brain.
That's wonderful. So using memory systems to understand memory systems.
Exactly.
So
a good way of putting it.
Can you give us an example of what you think is achievable in, say, 5 years compared to what is achievable in, say, 30 years?
Sure.
I mean, it's speculative, of course.
Yeah. I think over the next 5 years, I hope we'll get these systems working in live mice. And I think it's reasonable that we might be able to put a mouse in one environment and give it some cheese, and then put the mouse in a different environment and give it some Nutella, and then take out the mouse's brain and ask what was different about the representation of the cheese versus the Nutella, or something like that, to start to learn which patterns of which sets of neurons represent different subtle aspects of an experience or a memory in the mouse's brain. Now, going 30 years is, who knows if what we're going to be doing in 30 years. But I guess my hope is that with some combination of these kinds of tools and the kinds of tools that we've been developing, one will be able to really get sort of a complete picture of the biological macromolecules, DNA, RNA, proteins, as well as the small molecules and the signaling molecules and the ions that are sloshing around in the brain, and the sort of dynamics and the voltage all at the same time. And the dream of ultimately, of course, is not just to collect a lot of data, but to actually have some understanding of it. And I hope that over the 30-year timescale, we might finally start to be able to distill out of all of these giant data sets some of the principles which must be in how the brain is storing and processing information. But that, I think, is going to take more than just better tools. That's going to take improvements in conceptual understanding as well.
Yeah, that's crazy and worth enough pursuing this research. So let's take an example. What is your cellular level event that you want to monitor? What is your dream event, let's say? You've been dealing with big scale things like monitoring a living animal. Let's now think about the molecular level. Some very curious signaling or whatever that you really like and you want to monitor. Do you have an example, or just taking an example to show how things are done?
Sure. There's another project in my lab where we're looking not at the level of the whole cell, but we're looking at individual synapses. And one question is, can you make ticker tapes or ticker tape-like systems that work at individual synapses that will record when that synapse is active or what the history or plasticity is of that synapse? Because memories are perhaps not just represented at the level of individual neurons, but we think that they involve specific synapses getting stronger and weaker. And so we'd like to be able to record those histories as well.
Mm-hmm. And what do you think this technology might achieve in terms of diagnostics? Maybe is it worth it also to understand how diseases work and how a single patient disease is working?
Sure. Well, so I think there's really two parts to your questions. One is, can we use these tools to study diseases? And that's not something that I'm working on in my lab, but I do think that as the tools mature, that will be an important set of applications. There are excellent rodent models of some diseases. You can also use these tools in human stem cell derived neurons, which you can use to model different diseases. And you might be able to use them to identify cellular or molecular signatures of the disease, and then to test for different candidate therapeutics to try to see which can restore normal function in these cells and tissues. And that's, I think, a promising direction. A separate direction, which I think you sort of hinted at, is towards diagnostics, meaning in a person finding out what is causing that person's illness. Now, I think it's somewhat impractical or not very likely that we'll use these tools in the brains of live people.
I don't think so.
Because they involve genetic modifications and they're pretty invasive. It's conceivable that you might use these tools in stem cell derived models. So you can take some skin from anybody and take those skin cells back, reverse the process of embryonic development to turn them into then you can differentiate those stem cells into neurons, which you can grow up and you can do whatever you want with them, study them. And those neurons are genetically identical pretty much to the neurons in the brain of the person who gave the skin. And so to the extent that the person's illness is genetically driven, and to the extent that you can recapitulate it in a cell-based model, both of these are huge caveats most of the time, which are not really true, but in some cases they are, then there could be a hope that you might use these cellular models and a variety of different measurement techniques to the molecular basis and ultimately potential therapeutics for that person. That's, I think, a somewhat far off goal. But using these just to study basic disease biology, I think, is a very realistic and near term.
It's very valuable, yeah, indeed. Adam, thank you so much for answering all my questions related to this wonderful work. I have also a bonus question I'd be happy to ask you. You are a startup founder with Q-State Biosciences, and you are deeply involved in education and outreach, especially I read in Liberia. Can you tell us how a scientist can also be an entrepreneur and a teacher? How you understand that your scientific work holds commercial potential, and what's the beauty, the nightmare of founding a biotech? And yeah, last but not least, the best of what you've learned with your experience in Liberia. That's a lot.
Okay, well, that was a lot. That was a lot of questions in one final question. Okay, so briefly, yeah, about 8 years ago or so, I teamed up with a colleague, Kevin Egan, who was in the stem cell department here at Harvard, and with David Margulies, who was at Boston Children's Hospital, to make this company, Q-State Biosciences. And Q-State uses the voltage imaging, the optical electrophysiology tools from my lab, combined with the stem cell-based disease modeling, which I just described, including many technologies from Kevin's lab, to try to create human cell-based models of different diseases and to use the electrophysiology tools to characterize those models and to test candidate therapeutics. And it's been an incredible learning experience to try to take these ideas, which were, sort of somewhat vague ideas initially, and the prototypes from our academic lab, and to really refine and polish them so that they're robust enough for application in drug discovery, which requires a very high standard of reliability and reproducibility in order to proceed. And so, maybe this is obvious, but I've learned that drug development is really hard, and that a lot of sort of academic lip service towards, oh, maybe this could be useful for drug development is not always realistic. That when doing the drug development, it's really important to have really clean and sharp hypotheses and a very specific notion of how the tools will contribute towards information which can advance a drug towards use in patients. And it took us a while as a company to try to figure that out. I think that now the company is on an excellent path and has some very specific programs towards developing therapeutics for pain, where we have clear biological hypotheses and are moving molecules towards clinical applications. And so I'm very optimistic that this will eventually contribute towards better therapies for patients. And it's just amazing to me that these genes taken from a Dead Sea microorganism could, over decades, eventually be turned into tools which are used for studying human diseases of the nervous system. In terms of Liberia, when I was in high school, I mentioned I grew up in New York City, and one of my high school teachers and mentors was a man named Asumana Randolph, who had grown up in Liberia. When I was in high school, it was the time of the Liberian Civil War, and so there was tremendous bloodshed and suffering in Liberia. And he would spend time during the day collecting pencils and notebooks and other things that students had left behind in the hallways of the school to ship back to his relatives in Liberia in order for them to use. And out of his income as a public school teacher in New York City, he was supporting a school in Liberia. And so after I graduated, we kept in touch, and eventually I decided to, along with a friend, to go to Liberia to try to help out with some of his science education and outreach efforts. And so this was a while ago. I spent parts of 2 summers in Liberia working on trying to do teacher training and helping set up labs that people could do with the resources that were available locally there. There are a lot of challenges associated with doing education in this context. There's no reliable electricity, not necessarily clean water, and big problems with malnutrition, malaria, hunger, corruption, violence. There a lot of really hard problems there. And certainly one of the I really grew to appreciate from this experience was just my incredible good luck. The people that I interacted with in Liberia were every bit as smart and talented as people that I knew at Harvard or in New York City, but they had, by chance been born in a context where they didn't have the opportunities to have the same kind of education and to pursue the kinds of activities which I, basically by luck, had been fortunate enough to be born into having. And so, it was really a striking contrast between the situation in Liberia, where annual income is a few $100s, and the situation in my lab here where we'll spend a few $100s for a mirror without even thinking about it. And I think it's important to think about how one can try to increase opportunity. Not just in Liberia, but of course, even within the United States, of course, we have huge variation in the degree of opportunities that people have at birth.
That's wonderful. Thank you so much, Adam, for explaining to us everything about your research and your wonderful career. There would be so much more to ask you, but time is limited. So I only thank you so much for all the insights, and I wish you best of luck with all your efforts, really.
Thank you, Luca. Best of luck to you too. Take care.
You've just listened to A Biotech Futurist, a podcast by Luca Fusarbassini. This is the first series, and a new episode is out every Monday. Please consider subscribing and rating the podcast on Spotify, Apple Podcast, YouTube, Instagram, or your top podcasting platform. And if you liked this episode, consider sharing it with your friends, as the growth of new podcasts relies on word of mouth. If you have any suggestions, don't hesitate to reach out to me on Instagram or Gmail at thebiotechfuturistpodcast@gmail.com. You can find the full AI-generated transcript of this episode on my website, lucafusarbassini.com. I'll also post the links to the main papers referenced in this episode, which you can find here in the description too. Thanks for listening to A Biotech Futurist. I am looking forward to talking with you in a week.